Automated DNA sequencing. Now we have the idea that in manual sequencing, like in case of chain termination method, the DNA fragments that are variable in their sizes, they are separated on agarose gel, and then the separated fragments, they are detected by autoradiography. तो इस तरह की जो मैनुअल टेक्निक्स हैं ये ऑटोमेशन के लिए वर्क नहीं करती, बिकॉज इन केस ऑफ ऑटोमेटेड प्रोसेस द एनालिसिस ऑफ डीएनए फ्रैगमेंट्स इन रियल टाइम इट इज़ रिक्वायर्ड कि जब डीएनए के फ्रैगमेंट्स सेपरेट होते हैं एग्रोस जेल में, तो एट द सेम टाइम दे आर आल्सो detected. So this is the basic procedure in automated DNA sequencing. And here we are using different type of fluorescent dyes to label our dideoxy nucleotide triphosphate. So radio labeled material they are not used, rather different type of chromogenic materials like the fluoro-4 they are used to stain the dideoxynucleotide triphosphates. And suppose here in case of the Sanger method, the reactions they are carried out separately in 4 different reaction mixture, and after performing the reaction, then the different fragments that are variable in length, they are detected by using a specific detector. So all the fluoro-4 we are going to use, they are different for respective dideoxynucleotide triphosphate. Like adenine, it will have different chlorophore, and similarly different chemical dyes they will be used for other nucleotide bases. So spectrally the staining reagents that we are going to use, they will emit different colors that will be detected by using the detector. In addition to fluoro-4 or the staining dyes, some specific primers that are labeled with dyes, they can also be used. Now suppose we have performed a reaction which is called as chain termination reaction by using different labeled fluoro-4 dideoxynucleotide triphosphates, then all the 4 mixture they can be run within the single lane of the gel. So 4 lanes they are not required, rather the 4 mixtures they will be run on the same lane and they will be separated depending upon the length of the DNA fragments. So detector will detect the emission of different dyes, and each emission that have a particular color, it will belongs to a particular base pair. So in this way, by running within the single lane, the fragments they can be detected. So here different lanes they may contain different samples, so more samples can be run within a single slab of the gel. So this is one of the advantage of the automated sequencing. So this is the block diagram of an automated DNA sequencer. So it is here the slab gel, and it contains different lanes, and within a single lane one sample can be loaded. So 4 different reaction mixture they are loaded within a single lane that will be separated depending upon the length of the DNA fragments, and each dideoxynucleoside triphosphate it is labeled with its respective fluoro-4, so it will emit different color that will be detected by the detector. So here different samples they are run in their respective lanes, so more samples for sequencing can be analyzed, and each will be represented by different colors. For example, different color for adenine and thymine, it will be, I think you will better understand in next diagram, that each base, it has its own fluorophore. For example, for the thymine, the color is red. For the guanine, the color is black here. It corresponds to a black peak, and green it is for adenine. So different fluorophore are there for their respective dideoxynucleoside triphosphate. So whenever the chain reaction it is terminated, it will be detected on the single lane within the gel. Automated DNA sequencing has several advantages. As I told you that in case of manual sequencing like the chain terminator, the autoradiograph they are produced, and even some good laboratories they face problem because the quality of the autoradiograph that is produced, it is poor, and to read the different segments in comparison with the standard lane of the nucleotide, it is difficult. So here one can face problem that the sequence may be read incorrectly. So this is one of the disadvantage of manual sequencing, but in case of automated sequencing it is not the problem because there is a machine or detector that can read the sequencing. So we can say that in case of manual sequencing, a skilled worker it is required. Whereas on the other hand, another advantage is that in case of automated sequencing, all the work it is done by the machine or the detector. Next advantage is that in case of manual sequencing, we may require 4 different lanes for 4 different reaction mixture. But this is not the case in automated sequencing, because here all 4 reactions they can be loaded on a single lane. And now there are some sequencer where the capillary tubes they are used that may contain 48 or up to 96 capillary tubes. So we can analyze up to 96 samples, 96 different samples, so that one can analyze thousands of nucleotides within short course of time. So these are a few of the advantage of automated DNA sequencing.